When discussing medical treatments, we often think about the use of small molecule medications which follow a simple chemical structure. These are generally pill-based, such as warfarin, the anticoagulant. Next, there are biological treatments, consisting of complex large molecule proteins. These are injected or infused into the patient. Infliximab, often prescribed for arthritis or Crohn's disease, is a good example of this type. Finally, there are cell and gene therapies, which are also known as advanced therapies. So what exactly are these? Simply put, cell therapy is when whole living cells are administered into a patient to treat a condition, whereas gene therapy is when a new modified sequence of deoxyribonucleic acid or ribonucleic acid, that's DNA or RNA to you and me, is inserted into the body as an addition or to replace or repair a mutated sequence. Let's look at cell therapy first. Although we often think of it as a new approach, cell therapy has been around for hundreds of years. The oldest form is a blood transfusion, with the first successful human-to-human transfusion carried out in 1818 by James Blundell at Guy's Hospital. Blood was taken from a man to his wife who was suffering postpartum hemorrhaging. In the 1960s, it was recognised that healthy blood stem cells from a donor could replace radiation-damaged cells, aiding the recovery of patients undergoing aggressive cancer treatment. This therapy is now routine. Last year, more than 100 million blood transfusions took place worldwide. So how does this work? It's all to do with the unique ability of certain types of cells. Let's start with stem cells. Unlike normal cells, stem cells can develop into other types of cell. So a blood stem cell could become a nerve cell, brain cell, muscle cell, or any of the 200 plus tissue cells of the body. Normal cells, on the other hand, are known as specialised cells, which have a dedicated role and can only replicate itself. Lymphocytes are another type of useful cell. These are white blood cells which are part of the immune system. They can be B cells, which produce antibodies which attack invading viruses and bacteria. T cells have the ability to destroy infected cells. And natural killer cells live up to their name by killing off malignant cells. So we can see if these very industrious cells could be engineered to treat a specific disease or repair damage, the health benefits could be immense. Now let's move on to gene therapy. In the last few decades, genomics, the study of genes, has leapt forward. Technological advances now mean your entire sequence of DNA, known as your genome, can be read in a few hours. This allows clinicians to identify damage and mutated genes which cause disease. A specific response can be engineered by modifying a gene sequence. This new sequence is known as a transgene. But there is a problem when it comes to putting this transgene back into the body, as nucleic acid, the components of DNA, is rarely taken up by cells. Fortunately, there is a solution, if a tricky one. A special carrier, known as a vector, is used to deliver the transgene. A vector is usually in the form of a virus which has been treated not to cause illness. A lot of research has been undertaken to find the most effective and safest vector. Cell and gene therapy are often used in combination. Let's look into the field of immunology to illustrate this. As you know, T cells are an integral part of the immune system. One of their functions is to seek out and destroy harmful cells. However, a patient's own T cells find it difficult to identify some cancerous cells. In gene therapy, blood is taken from the patient and the T cells isolated from the blood. In a laboratory, the gene sequence in the cells is modified to spot cancerous cells. As this takes place outside of the patient's body, it is called ex vivo therapy. The modified cells are multiplied, their millions are re-administered into the patient by injection. These modified T cells are now able to spot the cancerous cells and kill them. Pretty smart, huh? Treatment like this, where the cells are harvested, modified, and then put back into the same patient, is known as autologous. The treatment is personally tailored to each individual. If the donor of the cells is someone other than the patient or a group of people, it's known as allogenic. So what about ATMPs? This stands for Advanced Therapy Medicinal Product. The European Commission defines this as either a gene therapy medicinal product, a somatic cell therapy medicinal product, or a tissue engineered product. So ATMPs encompasses gene, cell and tissue engineered therapies. ATMP is a term you're going to be hearing more of within your workplace. it may eventually be used more often than the terms cell and gene therapies. In recent years, the UK has attracted substantial funding and interest in its ATMP research, which means more products are being developed, calling for an increase in clinical trials. This is where the research community can make an impact. By engaging with these new treatments and undertaking clinical trials, more ATMPs can be accessible to those patients who really need them. It is an exciting time for advanced medicines. The difference they can make to the NHS and patients is incredible. Time to get active and get those clinical trials underway.